All right, hello, welcome to the DevoWorm meeting for this week. Today I want to talk about some data sets that we have available. It was from a paper that was published in 2016, some work on binarizing the lineage tree and turning it into what we call differentiation code. And this was by request of one of our GSOC scholars, Jayadratha. And then I will get into some papers that I have to go over for the group so they can see what's going on there. We had some nice presentations last Friday in the orthogonal lab and orthogonal research and education laboratory open source meeting. We've kind of moved those over there now because I want to have all of them together. So Jayadratha and Leylith Baru, they both have talked about their projects. So Leylith is working on hypergraphs and trying to figure out how to build hypergraphs more efficiently. And Jayadratha is thinking about time hypergraphs or temporal hypergraphs. So that's what's going on there. So if you go to our website, devoworm.weebly.com, we have publications. So if you go to more academic output publications, we have a lot of publications here for you to look over. This spans the over 10-year history of the group. So we've done work on, of course, C. elegans and nematodes. We've released data sets. We've released software. We've done papers on fruit flies, one paper on fruit flies exclusively. We've released some data on this. We've done some papers on diatoms, on the genera Vasculogenesis, but other diatoms as well. Then we've done comparative development. So we've done a lot of work on looking at not just C. elegans, but C. elegans in evolutionary context. So if you go down the line, so if you use other species as a comparison, we can say things about development that are, basically putting C. elegans in context. And C. elegans is its own unique system, but also has a common ancestor to some of these other systems. We can find out some very interesting information about development. And so this paper here from 2016, Quantifying Mosaic Development, this is a paper that Richard Gordon and myself worked on. And we have a lot of supplemental data in that paper. And quite frankly, a lot of ideas that we've kind of forgotten about because this was almost 10 years ago. This paper was sort of a comparison, or at least a dual analysis of C. elegans, so our nematode, and Ciona Intestinalis, which is a sea squirt, which is a marine invertebrate. So this was published in the journal Biology, Basel. And there's some supplemental data. So if you go to the supplemental data tab, it gives you this zip file. And in the zip file, we have these two files. It's table S5 and table S4. So if we look at the paper here, it says Quantifying Mosaic Development. And, we do this analysis of Ciona Intestinalis and Caenorhabditis elegans. So one is, of course, our nematode, and Ciona Intestinalis is a sea squirt. You might think that's quite a phylogenetic distance. How do you say anything about it? And one of the things about both of these systems is, first of all, there's a lot of secondary data that we can analyze. But secondly, they have a very similar mode of development. That is, they have this sort of deterministic mode of development early on, where you can trace the cells through a cell lineage tree, and this unfolds in a stereotypical way across different members of a species, and we actually have a good nomenclature for it. So these are two systems that we can compare, not necessarily always directly, but it gives us this framework for doing what we're going to do, many of the things we did in this paper. And so I want to highlight some of the things that we did in this paper. So let's start with table supplemental 4. This is a type of analysis that we did on the lineage tree. It's called the differentiation code. And so what the differentiation code is it's a binary code that, and we also did this to the lineage tree as well directly. What this is a code that describes the relative position in lineage and the relative position in sort of the age of the embryo. So if we start with 0, we start with a single cell embryo. That single cell divides into 2 cells, the left-hand cell and the right-hand cell. Left-hand cell being 0, 0, right-hand cell being 0, 1. In C. elegans, that lineage tree is organized anteriorly, posteriorly. In Ciona Intestinalis, it's organized in a different way, but it's very similar in that it's connected to the morphology, connected to the phenotype. And so as we move through the tree, we add in our divisions. We end up with these codes for each level of the tree. So if we go to the next level, we have 0, 0, 0 on the left, 1, 1, 1 on the right, and there are all the different variations in the middle. So as you know from translating binary to decimal, you know that 0, 0, 0 is 0, and 1, 1, 1 is 1, or is the highest number, and then you have things in between. So if you're familiar with binary and translating binary to decimal, you know that 0, 0, 0 is 0, and 1, 1, 1 is 4, and then we have intermediate states 0, 0, 1 and 1, 1, 0. So basically every time a cell divides, we flip a bit in one direction or another. We then count for that level, and we have our 4-cell state. We also have our 8-cell state, so if we start from 0, 0, 0, 0 on the left and 1, 1, 1, 1 on the right, we get 8 cells, and we have these intermediate states. The point is that we can characterize the length of a code, and it characterizes a level of division. It also describes differentiation because it describes if a cell differentiates where that is in the tree or where that is in the lineage. And so the differentiation code varies from the lineage code by being reorganized by I think in this case it's the smaller cell on the left, the larger cell on the right. And this work on differentiation code is based on Richard Gordon's work on differentiation trees and differentiation codes, basically reorganizing the lineage tree so that you have tissues of different sizes resulting in different parts of the tree. So you're basically reorganizing the tree by some significance criteria. And so that's the difference between the differentiation code and the lineage code. But this actually provides a means for us to characterize each entry in the tree and address it in a way that's computational. So one of the advantages of generating this code is that we can describe it not in terms of we can describe it in terms of sublineage, and we can also describe it in terms of its relative position. So if we decide to, say, knock out a cell, if we want to run a simulation and we have our lineage code or differentiation code, when we run a simulation, we have an addressable set of cells. We know kind of their location in terms of how we organize our tree. So the lineage code, if we have 0, 0, 0, 0, we know this is at the 3rd level of division. We know it's at the left-hand side of the tree. And then for the lineage tree in C. elegans, we know that that's the anterior-most end. And so we can figure out very quickly where cells fit in. And so we can remove cells and mutate the lineage tree and run experiments to see if the lineage tree is still intact or if it falls apart, if it results in a viable embryo versus an inviable embryo. Because remember, all of these developmental cells that generate this lineage code go on to differentiate. And if we knock out certain cells, we can say, because in C. elegans and also I think in Ciona, those original cells have to be completely there in order for the next stages of embryogenesis and development to occur. So if those cells are missing, then you have no chance to recover those. Now, in mammals, that works quite differently. You get plasticity, you get cells that divide in different ways and differentiate in different ways, so they can fill in the gaps. But in our what we call our mosaic development mode, this is not the case. So these codes are very useful for this reason. So this is table S4 is for Ciona Intestinalis. As you can see, it has a nomenclature, and the nomenclature is it's based on, not really sure what it's based on. I can't remember, but it's it's a little bit unwieldy unless you really know what you're doing. The point is that we can actually create a code for this, and if I were going to do this again, I think we'd probably add some other geometric cues to this, or you could even combine lineage code or differentiation code with different um, other information. So you'd have a string, which is the differentiation code. You might have another string, which is a geometric code, which describes the sort of the coordinates of the cell. And certainly in C. elegans, we have those data. We have the we could have any sort of coordinate system we want. A lot of this data that we have are sort of inherently Euclidean distances. You could have other projections as well, other coordinate systems. And of course, you can add this code to the coordinate system, so you can represent this computationally as a binary string and a tuple with the coordinates in the tuple and the binary string as this location in a tree and then a location in a volume. And so that's what this is. And so this makes it much simpler computational sort of processing here. And then we of course have the Hamming distance. So the Hamming distance is the distance from it's sort of the distance from the origin. So we have a number of Hamming distances calculated here. We also have the lineage depth, which basically is the same as the length of the binary code, which is not surprising since I already told you that this is every time there's a division event, we add in another bit to the string, and it's longer and you get more possibilities. If we go to table 5, this is actually for C. elegans. So now we can see C. elegans, how this works. We have nomenclature on the left, which of course we recognize AB and its sublineage, and then we have P1 and its sublineage, and all the cells in the developmental lineage tree. And so we have a few more measures in here. We have, of course, a Hamming distance, a composite Hamming distance, and of course a lineage depth. So here we start at AB and P1. So AB is 0, P1 is 1. And so we start from a little bit different starting point than in Ciona, but we can align these, I think, pretty well. And so we, as you go deeper into the tree, as you get division events, lineage depth increases, the length of this binary string increases. You can calculate a Hamming distance from the origin point or from any 2 points in this addressable system. Now, one of the things we did, and I'm not going to pull up this table, is we calculated something called a cast code. And so this is not something that I think is useful for hypergraphs, which is originally why I'm showing you this data. But the cast code is basically it's like a blast sequence analysis, where you take two sequences and you align them, and then you calculate a distance between them. So the cast method is where you take these two differentiation codes or two lineage tree codes and you align them, and they might be of different lengths, so you have to account for the gaps, and then you can use an algorithm to calculate the distance between them. And so this is another thing we can do with lineage tree data. Once every cell is addressable, then we can do a lot of this other stuff as well. Okay, so it looks like Morgan is here. Hello, Morgan. I want to go over a couple of papers that are in our queue, and I think those are going to be interesting for a variety of reasons. I'm not going to go too deeply into them, but I'm going to go through 3 papers, and I'm going to go through their abstracts and some of their significance to the group. So the first paper is, I think this is a review article, or it's an overview article in eLife, and this is something that Susan sent me, so I wanted to go over it a little bit more detail. This is the overview article for this paper evaluating the transcriptional regulators of arterial gene expression by a catalog of characterized arterial enhancers. So this is a deeply molecular paper. So why is it that we're talking about it in our group? And that's because it helps us understand endothelial cells. So these are basically what determines whether an endothelial cell becomes part of an artery, a vein, or a capillary. So this is where you get cell differentiation in the heart, where you get these endothelial cells and they become part of the morphology, they differentiate. So and of course, Susan's work on epithelial cells, we have these epithelial sheets and we're looking at the physics and the mechanics of the sheet. And of course, that leads to gene expression and that leads to the integrity of those sheets. In this case, we have a different, we have endothelial cells, and we're thinking about how those cells in say a mammalian system become differentiated. And so they kind of talk about the blood vessel system. So the blood vessels in our bodies are either arteries, veins, or capillaries. Arteries transport blood away from the heart, and veins carry it back, and capillaries connect arteries and veins, also facilitating the exchange of gases and nutrients between the blood and different organs. The inner layer of every blood vessel is formed by endothelial cells, but what factors determine whether a newly formed endothelial cell becomes part of an artery or a vein or a capillary? And so this is about the formation of blood vessels during embryonic development, and this is where it begins with the differentiation of mesodermal cells into endothelial cells. So we have one of the 3 germ layers, and those cells from the mesodermal cell layer basically differentiate into endothelial cells. This allows for new blood vessels to be generated through a process known as vasculogenesis. And so there's a citation, Choi, 1998, that talks about vasculogenesis and reviews that system. Subsequently, additional blood vessels arise from pre-existing ones via angiogenesis. Although the first arterial endothelial cells are produced during vasculogenesis, studies performed on humans, mice, and zebrafish have shown that most arterial endothelial cells originate from venous endothelial cells. And so the differences between arterial, venous, and capillary endothelial cells has become the subject of much research, but the substantial changes in gene expression that are required for venous endothelial cells to become arterial endothelial cells have received less attention. So this new paper in eLife here, cited Neurons et al., report that they've identified a list of genetic enhancers and transcription factors that have a crucial role in this process. Enhancers are non-coding regions of DNA that are involved in regulating gene expression, while transcription factors are proteins that regulate gene expression by binding to enhancers. And so this goes through kind of what they do here in the paper, and they find 41 putative arterial enhancers, 3 of which have been previously studied in vivo. Subsequent experiments on genetically engineered zebrafish that used green fluorescent protein. So this is a marker that you can see under a fluorescent microscope, and it tells you sort of its presence in the thing that you're trying to look at. It's basically used to report on gene expression, but also the presence or absence of certain types of cells, labeling those cells and their nuclei to localize, or it could be, other parts of the cell to localize this GFP signal and tell you if something is where you need it to be. Showed that the 16 of the enhancers drove gene expression in blood vessels, with 15 specifically driving gene expression in arteries. Moreover, the researchers identified at least one enhancer for each of the 8 genes. Further experiments on embryonic mice showed that these enhancers also drove gene expression in arteries. And so these experiments were done in both zebrafish and mouse. And so they were able to successfully identify a cohort of enhancers capable of directing gene expression to arterial endothelial cells. Then they also explored potential transcription factors that might bind to these enhancers. These transcription factors, such as ETS, SOXF, and FOX, are enriched in arterial endothelial cells, although they can also be found in venous endothelial cells. The researchers also found that the transcription factor MEF2 binds to enhancers in a subset of arterial endothelial cells associated with angiogenesis. And then the binding of another transcription factor, RBPJ, suggests that notch signaling pathways play a role in regulating the expression of arterial-related genes. So this has this complex interplay between general developmental mechanisms and some of these specific genes for the cell type. So this paper provides insight into regulatory mechanisms that define what they call arterial identity and the identification of various arterial enhancers and transcription factor binding sites for these enhancers. So, when we think about cell differentiation, we think about a cell and it's differentiating into some state. It could be a neuron, it could be a arterial cell. And a lot of times in what we call regulative development, we have what we call a cell identity or a tissue identity. And that is that the cells aren't unlike in C. elegans, the cells aren't deterministic with respect to their fate. They're cells, stem cells and progenitor cells that can, basically be moved to a number of different fates. It really depends on their microenvironment and where they're located, with who their neighbors are. So there are all sorts of experiments where people have transplanted stem cells or precursor cells into different tissues, and those precursor stem cells, those pluripotent cells, take on the identity or the phenotype and the molecular properties of the surrounding tissue. So this is something that people are very much interested in from a regenerative medicine perspective. How do we get these cells to sort of better integrate into tissues? Can we coax them in some way? Can we deliver some sort of molecular cocktail? Can we, identify cells that are maybe more integrated or less integrated into their host tissue? And, basically, how does this process work? So you notice I've not mentioned anything about mechanics of the cells. And so this is interesting because this sort of work, they talk a lot about molecular mechanisms, that they don't really talk about the mechanical aspects of different types of, how these cells are integrated. So going back to our example from epithelial cells, we know that these epithelial sheets exist in this sort of mechanical world where the mechanics are very specific to keeping the cells in place, even though they're sort of migrating and moving against one another constantly. And they're also triggering a lot of molecular mechanisms. There's a lot of sensory information each cell is providing to its neighbor, and there's all sorts of transduction of these signals, and it triggers a lot of transcription factor activity. And so it's very interesting how, they don't talk about the molecular aspect or the mechanical aspect of it. But it's very interesting to think about studies like this in terms of the mechanics of the tissue that we're looking at. So this looks like, I mean, I didn't want to get into the full paper because this is something that, is probably beyond the understanding of the group, and maybe my understanding of some of these techniques. This next paper is something from the bioRxiv. This is an older paper, but it's something that I found in kind of researching things. And this is talking about the developmental hourglass. And so this is an idea that goes back in developmental biology and in Devo to this notion of there being a sort of a phylotypic stage of development. So this is not in C. elegans, but this is in this regulative developmental mode, where you have a lot of variation before the phylotypic stage. Then you have the phylotypic stage, which is very regularized, and then you have the post-phylotypic stage where embryos will diversify into their different morphologies. So basically you have this stage where the mechanisms of embryogenesis are very stereotyped and very similar, and that's kind of in the middle of the process, hence the term hourglass. So if we know what an hourglass is, we have this top chamber and this bottom chamber, and they're connected by this narrow passage, and usually sand goes through the hourglass, and the sand is sifted through this narrow chamber, and that's the phylotypic period, is this narrow chamber. So in this paper, they're actually thinking about this hourglass, which is a metaphor that people use in development. But instead of just thinking about it as a metaphor, they actually do a computational model and things like that. So they use dynamical systems theory to get at this problem. Determining the general laws between evolution and development is a fundamental biological challenge. Developmental hourglasses have attracted increased attention as candidates for such laws, but the necessity of their emergence remains elusive. We conducted evolutionary simulations of developmental processes to confirm the emergence of the developmental hourglass and unveiled its establishment. We considered organisms consisting of cells containing identical gene networks that control morphogenesis, and then evolved them under selection pressure to induce more cell types. By computing the similarity between the spatial patterns of gene expression of two species that evolved from a common ancestor, a developmental hourglass was observed. So this is where they're looking at the spatial patterns of gene expression across the embryo. They're looking at different cells from anterior to posterior end of the embryo. They're looking at these gene expression patterns, and they're looking across phylogeny. So they're looking at two species separated by phylogenetic distance, but having a common ancestor. And so we can see this developmental hourglass, which is defined as a correlation peak in the middle stage of development. So what does that mean? That means that in the middle stage of development, there's this correlation between the two embryos, whereas earlier and later in development, we don't see this correlation. And we can assume that in the earlier and later stages of development that there's been divergence, phylogenetic divergence, but in that middle stage, things are essentially conserved. And so this is perhaps important for in terms of regulating different physiological processes and so forth. The fraction of pleiotropic genes increased, whereas the variance in individuals decreased. So you actually start to get these pleiotropic genes turned on, which means that you have genes that have multiple effects. You see pleiotropy in their outputs. So you get basically, you're getting sort of transitioning to this later stage of development where you really get this difference in phenotypes. But you also see a variance in individuals decrease, and this is consistent with previous experimental reports. Reduction of the unavoidable variance by initial or developmental noise, essential for survival, was achieved up to the hourglass bottleneck stage, followed by diversification in developmental processes whose timing is controlled by the slow expression dynamics conserved among organisms sharing the hourglass. So This study suggests why developmental hourglasses are observed within a certain phylogenetic range of species. So you see these hourglasses within certain clades within certain parts of the tree of life. And so it's very, it doesn't really give you a lot of insight from the abstract, but it basically gives you this, I guess in this case, we're reviewing sort of what a developmental hourglass is and what it produces. So the validity of the developmental hourglass model was first noted based on the coincidence between the phylotypic stage of vertebrates and the period of temporal and spatial progression of the Hough genes. It's in these developmental systems. You have Hough genes, which define the segments of the organism, which show up in terms of striping patterns. So you see this in Drosophila. You have very famous Hough genes experiments in Drosophila, showing that there are these segments of embryogenesis where you can define these different sort of stripes of phenotypic differentiation. There's the spatial progression, so it's spatially explicit. And then there's this phylotypic stage of vertebrates, so this stage where there's this sort of typicality. And so there's this connection between the two, and that's the idea behind the developmental hourglass. So this has been characterized for various taxa by bioinformatics analysis over the past 2 decades. We see this for chordates, for echinoderms, for arthropods, for nematodes even, for mollusks, and also suggested beyond the animal kingdom for flowering plants, for fungus. So you see this across development, you see this across the tree of life. You see this in different parts of different taxonomic groups, and so there are all these reviews on this. One may state that the observed data support a developmental hourglass, at least in some phyla. So there's variation on this interpretation of the data. Despite the growing evidence for the developmental hourglass model, whether it's just a contingency in evolution of multicellular organisms or a necessity therein, is still in question. In other words, the possible mechanisms underlying the evolutionary acquisition of mid-embryonic conservation remain elusive. Recent experimental studies have concluded that the developmental hourglass is not sufficiently explained by fragility in the bottleneck stage, but rather developmental constraints or limitations on phenotypic variability are suggested to be the main driving force in the emergence of the developmental hourglass. The relevance of self-organizing processes to the formation of the hourglass was discussed in this citation 26, whereas there is no consensus on the mechanism for developmental hourglasses. So this is a schematic in their simulation. So what they did is they took development and they used a simulation, an abstraction of this. So in this case, we go from A to B to C. In A, which is here, we basically have each individual, which is a unit of selection, contains cells aligned in a one-dimensional space. These contain the same gene network. The state of each cell, given by the gene expression pattern, changes according to identical gene networks. There are 100 individuals in the evolving population each generation. So they have this sort of population-based model in A. In B, starting from the initial states, the gene expression patterns change according to the gene regulation network. After sufficient time, the expression of each cell reaches the terminal state on or off. Cell type was determined according to the on-off patterns of the target genes. For the 4 target genes, there were 2 to the 4th possible states. So here we have this initial state and then development, and we have these different states that are colored, and then we have the terminal state. So basically, we go from the initial state to the terminal state, and there can be this sort of up to the terminal state. And then C, which is here, so it's this, simulating of regenerations. The selection procedure of species diverged from a common ancestor. And so the left chain represents the results of the first step of the evolutionary simulation to select one species. So we have this first step where we have evaluation at every 200 generations. And then the second step is where an ancestor at a certain generation was cloned, and additional evolutionary simulations were carried from the ancestor up to the same generations of the original. So they took every 200 generations, actually, it starts at 400 generations here, they took a clone and evolved it alongside the original. So they were doing this sort of clonal evolutionary simulation. And so they conducted this evolutionary simulation to investigate the mechanism behind the establishment of this developmental hourglass. They adopted a dynamical system scheme to separate spatial patterns, which is commonly adopted in theoretical studies. The model for the simulation can represent cellular states that are determined by gene expression patterns, governed by gene regulatory networks and spatial cell-cell interactions, leading to the differentiation of cell types and pattern formation. So this is where we want to model this sort of diversity of things that allow for differentiation. It's basically, you can have one-step differentiation, multi-step differentiation, but there are a lot of things that contribute to each type, each stage of differentiation. It's it's not just simply flipping a switch, although that's often the way it's sort of portrayed in the literature. You have all these things kind of leading up to it. Individuals must be selected according to a biologically relevant fitness function. We assume that if there are more cell types within an individual, they attain more functions, such as the fitness is higher. The fitness function reflects the idea that different cell types have different cellular functions, so that species with many cell types can deal with multiple tasks. Hence, they are more adaptive than those with fewer cell types. Each cell type was defined according to the on or off state of the given in output genes. In other words, our simulation mimics competition for survival, where individuals acquiring as many cell types as possible are the most adaptive and would leave more offspring. So this is an interesting kind of developmental simulation that's driven by this sort of evolutionary simulation. And it's really interesting the way they set this up. Now, to study the developmental hourglass, the developmental processes of extant species that diverge from a common ancestor must be compared. So we have this sort of comparison between two different systems. Thus, the model must represent not only a mother-to-daughter single-chain pedigree, but also must be able to examine a phylogenetic tree that contains multiple pedigrees as branches. So this is why they did this evolutionary simulation in figure 1C. They wanted to take basically from 0 to 400 generations, you have this common ancestral state. And then at the second step, you start to get these clones that also evolve in parallel. And so you end up with these things you can compare your system to. So they did this two-stage evolutionary simulation. In the first stage, simulation of one lineage was performed to obtain one species by selection. In the second stage, evolutionary simulations were executed over the same number of generations using an ancestor in the lineage of the first step simulation. Using different random numbers for mutation, extant species branching from the common ancestor were obtained. And so then using the above setup, we aim to examine the correlation between gene expression patterns and the development of multiple species that diverged from a common ancestor. The results showed a developmental hourglass in the sense that the peak of such correlations lie in the middle of development. We also confirmed the other reported characteristics, including an accumulation of expressed pleiotropic genes and reduction of intraspecies expression variants at the developmental bottleneck stage. The developmental period of the hourglass bottleneck obtained in the simulations was determined by the genes whose expression changed slowly during development, acting as genes to schedule development. So this is this scheduling process where you get this change in gene expression over development, and there's this sort of schedule to it. The bottleneck serves as the developmental foundation that sufficiently attenuates the effects of initial noise to prepare for subsequent developmental processes. Based on these results, the validity of developmental hourglasses and possible mechanisms for its emergence were discussed. So they use what they call a GRN, or gene regulatory network model, where you have this network of genes and they have this signal that kicks off the cascade, and you have these different genes that are upregulated or downregulated, and then it affects the next partner in that network. And you continue through the network until you get these products that are the outputs of the network, and then the products of that network diffuse through space. So you have this sort of, I guess you could call it a spatial GRN that they're using. And so then they run this simulation until they reach a fixed terminal state after sufficient developmental time, where the expression of each gene in the network is reached an on or an off state. Depending on the combination of on-off expression patterns of the given output genes, a cell type is determined. And so in figure 2a, they show specific examples of the developmental dynamics of two individuals acquired from evolutionary simulations after 1,000 generations. And so this is in figure 2. This shows examples of the evolutionary simulation that showed hourglass-like developmental correlation. And so these are very colorful graphs. You have A1, A2, and then B and C. So in A, we have A1 and A2. So these are two examples of gene expression dynamics of evolved genetic regulatory networks. So this is one genetic regulatory network. This is the second one. So this is developmental time from 10 to 1,000. And then this is the cellular index, which they define in the paper. And then this shows sort of a phase portrait of that. This is different for these two different examples. This is presuming, I guess, two different species and how those behave, those taxa with phylogenetic distance. The idea here is that we have these spatial pattern transitions that we need to identify. We can see that these things change. They're more frequent in the early developmental time periods. This is consistent with experimental observations. Thereafter, the developmental time was plotted in logs. Following evolving 800 generations, the two GRNs evolved from a common ancestor of the 800th generation and then simulated further over 200 more generations, while suffering from different genetic changes, such as that the two have diverged from a common ancestor. Okay, so that's the first one. And then, of course, we have, so this is actually, let's see, B is here. So this is B, where we have developmental time of A2. So this is A2 here. And this is developmental time of A1 on this axis. This is A1 here. So basically, we're comparing A1 and A2. Well, we're doing this with a heat map. So we can see that there's this sort of distinction between A1 and A2. So this is figure B, the heat map of the similarity indicator, comparing the developmental dynamics of the two individuals in A. As we have developmental time of A1 and developmental time of A2. Both axes are in a logarithmic scale. Each point indicates the similarity of gene expression states between the two examples at specific time points. As the time scale of development can be different between the two in general, the comparison at different time points is included. And so the highest level of similarity appears to be in the middle stage, which is this area of the heat map here. And the green line indicates the locations where the similarity indicator takes its maximum for each developmental time step of individual one. So this is here, this green line, putting out like that. So I think I said that maybe the green line was something else, but that's not true. This is just each point in this simulation, so this entire phase portrait is just basically the comparison. Okay, now C, of course, then is the projection of the similarity indicator of the green line in B against the developmental time of the first species. So this is the green line here, and then there's this developmental time for A1, and we have developmental time for A1 here, and then this green line restated as this developmental correlation. So you can see that there's a correlation here in the middle period, the phylotypic stage, and then down here we have very low similarity later because things are diverging. Those two species are diverging away. in before the phylotypic stage, we have a little bit higher level of correlation, um, but not much. So the phylotypic stage is where you have this enhanced correlation between two species. Okay, this looks like, there are a lot of other results in here, and I'm not going to get into them. There's a lot of cool simulation in here and looking at the results. Sometimes these simulation results are hard to interpret because you have to live in the world of the simulation and know what's going on. So, and then this just shows like the steps in the evolutionary simulation, and again, more information about developmental correlation. So basically, they're playing around with different ways to do this, but you see again this developmental correlation in the middle of the simulation. The last paper I'm going to talk about here is this paper by Michael Levin and colleagues in the Saturday morning neurosim meeting that we had on Saturday in the Orthogonal Research and Education Lab. We talked about another Michael Levin paper. It's actually from, I think, a lot of the same authors on these biobots or these anthrobots and their transcriptional profiles and things like that. Is an interesting paper. This is a different paper, but it's very similar. Is this the same paper? No, it's not a different paper. The running title is Biobots with Neurons. So this is where we're looking at these biobots instead of anthrobots. We're looking at, these organoids with neurons, and they're using these bioengineering techniques to generate these artificial real organisms. Or I guess they're not real organisms, but they're biological in the sense that they're cells, but they're also they're constructs, I would say, is probably the best way to put it. So this is self-organizing neural networks in novel moving bodies, anatomical, behavioral, and transcriptional characteristics of a living construct with a nervous system. And so if we look at the abstract, they mention a great deal is known about the formation and architecture of biological neural networks in animal models, which have arrived at their current structure-function relationship through evolution by natural selection. Little is known about the development of such structure-function relationships in a scenario where neurons are allowed to grow within evolutionarily novel motile bodies. Previous work showed that when a piece of ectodermal tissue is excised from Xenopus embryos, allowed to develop ex vivo, will develop into a three-dimensional mucociliary organoid and exhibit behaviors different from those observed in tadpoles of the same age. These biological robots, or biobots for short, are autonomous, self-powered, and able to move through aqueous environments. Here we report a novel type of biobot that is composed of ciliated epidermis and additionally incorporates neural tissues. These are neurobots. We show that neural precursor cells implanted within the Xenopus skin construct. So this is a frog model embryo, and they have these skin constructs. So what they're doing is they're putting neural precursor cells into a tissue of skin, and it allows these, or they're able to get these neural precursors to develop into mature neurons and extend processes towards the outer surface of the bot, as well as among each other. So what they're doing here, and we talked about this in the first paper, when you put different precursor cells in a tissue, it responds to the cues, local cues of that tissue. So we could very well have, we might not be able to achieve, fully mature neurons in a skin construct. It takes a lot of guidance in terms of, how these cells are implanted and how these cells are able to develop. And so this is where we're trying to develop not just a single tissue, but a compound tissue that resembles an organism. So we have neural precursor cells in a skin construct. We are able to get mature neurons. We're able to get processes extended to the outer surface. So we have the system where we have a layer of skin with sensory cells in it, basically. We show that neural precursor cells implanted. Okay, we have that. These self-organized neurobots show distinct external morphology, generate more complex patterns of spontaneous movement, and are differentially affected by neuroactive drugs compared to their non-neural counterparts. Calcium imaging experiments show that neurons within the neurobots are indeed active. Transcriptomic analysis of the neurobots reveals increased variability of transcriptional profiles, expression of a plethora of genes relating to nervous system development and function, a shift towards more ancient genes, and upregulation of neuronal genes implicated in visual perception. Now, we talked about the accompanying paper on Saturday, so I refer you to that discussion. But basically, they found the same types of things. So it was a different experiment, but they found similar things. You get with these neurobots, you see, of course, increased variability of transcriptional profiles. So this is where we don't necessarily have a phylotypic stage here, but we're trying to construct different types of complex tissues from basically from scratch. We're trying to engineer them, and so we get these sort of variable transcriptional profiles, which suggests that you have this differentiation of cells that are mature and that are stable. Although it isn't always stable if you have a variable transcriptional profile, but that's what it kind of suggests. We get the expression of genes related to nervous system development and function, which means that it indeed is that these neurons are indeed differentiating down a neural pathway and not some other pathway, and that they're mixed in with like skin related genes and things like that. A shift towards more ancient genes, which is interesting because I think in the last paper on Saturday we mentioned there was a follow-up item, which is they talked about like sort of the phylogenetic origins of some of these programs. So like, this shift towards more ancient genes is basically, you get these, I don't really know what they mean exactly, but there's this connection to phylogenetics that's really interesting. And upregulation of neuronal genes implicated in visual perception, which I'm not really sure why that would be, but there may be that there are these genes for perception, in sensory neurons that are maybe, relevant here. But in any case, it's a very interesting set of experiments. They're trying to build these bots from biological tissues. They're trying to construct these complex biological tissues, and they're having some of the same problems that you see in development. So it's quasi-development, really, but you have a lot of the same issues. Okay, so this maybe explains more. Sensing cues from the environment and translating them into appropriate responses is the fundamental function of the nervous system in all animals. Critically, nervous systems endow animals with the ability to generate context and experience dependent changes in their behavior. So this is where we have, plasticity based on experience dependence or context dependence. Basically, when you put cells or you put sensory systems into a new environment, they change how they work, and so they exhibit plasticity. So this is kind of at the heart of what we're trying to do with these biobots. They're trying to figure out what the limits of plasticity are. So nervous systems are also known to be plastic and adapt both structurally and functionally on a much shorter time scale to changes in sensory and or motor effectors that might occur in the lifetime of the organism, say for example as a result of injury, amputation, or sensory deprivation. The degree to which the nervous system of an animal can adapt to a new body plan is especially impressive in cases where changes in sensory motor architecture are particularly drastic. So the ectopically induced eyes in the tail of Xenopus tadpoles were shown to confer light sensitivity to the eyeless hosts. So this is an example of where you have sensory cells, you have these eyes that are induced in the tail of the Xenopus. This is not a place where you usually see eyes, but they can actually confer light sensitivity. So they basically function as photoreceptors, just like photoreceptors in the eye, but they don't really give any visual input to the brain. They just give this ability to sense light. And so you might have this pathway that's constructed from that, where you get light sensitivity in different parts of the body. Our pineal gland in humans is like that, where you have the pineal gland was at some point in evolution a light sensing organ, and then in humans it's sort of evolved to be this pulsatile organ uh, for hormones. Okay, so what are the limits of neuroplasticity of nervous systems? How would wild-type neurons establish pattern and function in a new embodiment? So basically, we're taking neurons, we're putting them in a new embodiment, in this case, a layer of skin cells, and we're getting them to behave in a certain way. Will they behave in the way we intend? And so we want to create these truly novel configurations of biological materials, which allow us to probe the plasticity of evolutionary hardware, to adapt on developmental but not evolutionary timescales, to truly novel circumstances, which have applications for regenerative medicine, human augmentation, and biological engineering. We sought to establish a model system in which we could study the morphology and function of neural networks in novel scenarios, better understand the evolutionary developmental biology of nervous systems, and improve the design of future innervated constructs, meaning that you have cells in other cells and that there are nerves that come out of that and through the whole construct. So they point to sort of the history of this, where you have neuromuscular assembloids that have been used to model corticospinal motor functions, and neural cultures that have interfaced with computers or robots using microelectrode arrays to learn and perform various tasks. And these are, of course, hybrid robots that were not built exclusively using biological tissues, not fully embodied and not self-motile. So there have been people who have done these kind of biohybrid robots and other types of hybrid robots. And the challenge here is to improve upon these. Biohybrid bots that move through neuromuscular actuation have also been built by embedding neural and muscle cells in silicon polymer scaffolds. Similarly, such bots do not self-assemble, require external stimulation for propulsion, can only move at very slow speeds, have a very limited behavioral repertoire, and still require synthetic scaffolds. And so what they have here is they have tissue, ectodermal tissue that's been excised from an animal pool of a late-stage blastula stage Xenopus embryo. So we have sort of what we would call early development before the phylotypic stage. And we're taking ectodermal tissue from the ectodermal layer, and we're using that as a way to model self-motile, self-powered 3D musculociliary organoids. And so, yeah, we basically have these 4 cell types that are normally present in a tadpole skin: multiciliated cells, mucus-secreting goblet cells, ionocytes that regulate ionic homeostasis of epidermis, and small secretory cells. So these multiciliated cells can act as motor effectors in these organoids and can sense flow, and this arises from their polarized beating, and this can generate a suite of stereotype movement trajectories and velocities. Interestingly, small secretory cells were shown to secrete serotonin, which stimulates an increase in ciliary beating frequency through serotonergic receptors expressed on these multiciliated cells. These self-motile mucoscilatory organoids, which we refer to herein as biobots, are capable of navigating aqueous environments, generating a suite of stereotype movement trajectories and velocities. So this is really interesting. They're using this sort of system. It's sort of a derivative of Xenopus embryos, and they're able to produce these 4 cell types in the skin. But more importantly, they're able to establish this sort of sensory motor system, very primitive sensory motor system, where you have an embodied sensory motor system and then you can introduce neurons into the mix and create neurobots. So they have some really interesting results here. I'm not going to get into those, but yeah, I'd like to point people to that discussion on the Orthogonal Research and Education Laboratory in last week's Saturday meeting, where we talk about the other paper from this group on anthrobots. Morgan, are you available? I don't think you're available to comment on this, but it'd be kind of interesting to hear from you.
I'm afraid I have been multitasking, but diving into the kind of literature review that we're doing for 3D bioprinting, and the work that came up in terms of what Zev Gartner is calling 4D bioprinting, right? So it's 3D plus development, right? And it's just made me want to dive back into the anthrobot literature and the papers around that work, and the kind of mechanisms and kind of like the physics that's important in these considerations, or what's driving the cell's multicellular organization, right? So around the kind of thing that you posted on the engineering for life, or what was the Dresden meeting that you posted?
Yeah, it's like the mechanics of life or something like that.
It's something like that, right? Yeah. And again, there's the kind of stuff that comes up. I mean, I didn't necessarily see the Morpheus group in that, but Dresden has that Morpheus project where again, like tissue organization, especially in early stages, being driven by cell contact, and these kinds of things that only make sense when you actually do the simulations.
Yeah.
of how you can how much that self-organization is driven by cell adhesion differences, for instance, right? And so it's great stuff, and just wanted to see if you had a chance to look at that. The video from Arias is just another nice, really a researcher giving a wonderful overview of development.
Okay.
The Barcelona guy.
Oh yeah.
Anyway, but great stuff, and I'll try and review better when I'm, when it's not. It's school's over at this point, but there's still plenty of family activities at this time.
Yeah. All right. Well, thanks for showing up and listening in. Yeah, so that's all for today. Thanks for attending. Talk to you later.
Take care.
Take care. Bye.